Right, let's share screen again. Okay, let's just expand that. Right, good. Okay, so we've come to the second lecture following the nature of immunity lecture, covering the basics of the immune response. So this second lecture is on the molecules of adaptive immunity. And these are the antibodies. And as you can see from these structures here, you can see how wonderful and wonderfully complex these molecules are. First of all, the antibody classes or isotypes, IgG, IgE, IgD, IgM, and IgA, and the IgG subclasses, G1, G2, G3, and G4. And you'll notice on these diagrams here, we have these sugar chains, oligosaccharides coming off, called glycans, and that applies also to the other classes of antibody, E, D, A, and M. So these molecules are, antibody molecules are themselves glycans or glycoproteins. And the sugar plays a very important role, as we will see. Right, so the molecules of adaptive immunity, antibodies. So we're going to consider antibody structure and antigen binding. We're going to consider antibody isotypes, classes, and also the subclasses of IgG, and the basis behind antibody diversity. So we start off with antibody structure and antigen binding, and we'll use IgG as the main example. And there's a diagram, classic Y shape that you see in textbooks, and I'll point about that in a second. But the major antibody in the blood is immunoglobulin G, or IgG. And antibodies are made from 4 polypeptide chains, 2 identical light chains and 2 identical heavy chains. So these are the light chains in blue, and these are the heavy chains in red. And we have 2 Fab regions, fragments, and 1 FC region, fragment in an IgG molecule. It might vary with different classes, particularly IgA and IgM, but that's the basic IgG structure. Fab stands for fragment antigen binding. And this is the part responsible for antigen binding or epitope recognition. It contains the variable region between IgG molecules with contributions from the light chain, or each light chain, and each heavy chain. So this and this forms the antigen binding sites of the IgG molecule. Fab with a prime means Fab plus the hinge region, which contains disulfide bonds, and the disulfide bonding is through cysteine residues on the polypeptide chains. They contain the sulfhydryl group, and these can combine together to give a disulfide bond, which holds the two heavy chains together, and you can get a number of disulfide bonds depending on the antibody isotype and IgG subclass, again, as we'll see in a minute. And you have disulfide bonds holding the heavy chain and light chains together. So the numbers of disulfide bonds in the hinge region and length of the hinge region depends on the isotype and subclass. FC stands for fragment crystallizable, and when the structures of antibodies were being worked out half a century ago by Porter and others, it was soon realized that the FC fragment was crystallizable. In fact, the Fab fragment is also crystallizable. And the FC fragment, this is the same, or approximately the same, with some minor variations in all IgG molecules. Okay, it's worth pointing out, as I said, the number of disulfide bonds depends on the isotope subclass. And whereas the Fab regions bind to the antigen binding site or sites, the FC region binds to an effector molecule or receptor. Could be on some sort of phagocytic cell, but the FC region binds to an effector molecule or receptor. Now, although in textbooks the molecule is depicted as a Y shape, it's important to realize the antibody molecule is not a static Y shape in one plane or coplanar, but the three arms of the antibody, stand back a bit so you can see me, okay, can be non-coplanar, a bit like a traffic warden. So bear in mind that antibodies are not necessarily coplanar. The domains can lie in different planes. And as I mentioned before, these molecules are glycoproteins, and they're glycosylated on the FC region, usually 100% glycosylated on the FC region, and sometimes on the Fab region as well. And you need to see the Roy Jeffress lecture, which follows this one, which goes into glycosylation in some detail. And Roy Jeffries is I think it's fair to say, still the world authority on antibody IgG glycoforms. And the glycosylation is so important in terms of antibody function, both in defense as part of the immune response system and as part of antibody-based therapy. So remember that. And this is the glycan chain on the IgFC fragment, represented in this color and shape notation. So on the FC fragment, the glycan chain, sugar chain, is linked to the protein, heavy chain, via ASN 297. That's asparagine residue, which has a nitrogen on the side chain, so it's N-linked, asparagine residue 297 on the heavy chain. And this links to the red residue here is glucnac, which is N-acetyl glucosamine. This is fucose, which is a very hydrophobic saccharide residue. So these are sugars. They've got the same sort of ring structure, or similar ring structure like glucose has. And don't forget, there's a ring oxygen in all this, but with different functional groups coming off. So fucose has got a big methyl group, which makes it a very hydrophobic carbohydrate residue. Okay, this is mannose, this is galactose, and this is sialic acid. And sialic acid has this charge group, this acidic group on the end. As I said, the carbohydrate residue of antibodies and the glycoforms play a very important role. Right, now this concerns the effector function of antibodies via the FC region. So after the antibody has recognized the antigen on a bacterial cell or some other invader, then antibodies will gang up and they'll complex with the invader via the Fab domains, and then the FC domains will bind to the appropriate receptors on a phagocytic cell or whatever, and these will destroy the invader. That's opsonization. And there's a link to this video clip here. Now, in terms of antigen recognition, because the antigens, the amino acid chain or whatever, of a peptide fragment or whatever is left, or whatever is the signal which is being picked up as the antigen. Now, this may present itself in different conformations depending on the state of denaturation or otherwise of the peptide chain. So it's important that different conformations are recognized And this, if you like, represents different epitopes which are picked up by the antibodies. So epitope equals the antigenic determinant, and it could differ by different parts of the amino acid chain or even carbohydrate presented by the invader and the different conformations. It's important the antibodies can pick this variation up. Antibodies can be cleaved at various points by proteolytic enzymes, such as papain, to create various fragments. So you can cut the antibody at different points depending on the enzymes used. And this is often useful in the design of some antibody therapeutics. For example, this dye Fab containing two Fab fragments still linked together by the hinge region, but with the Fc domain cut off, that could be linked through some drug effector molecule. The FD domain, FD binding domain might be useful. So there's been a lot of research on the usefulness of these fragments. As I said, their smaller size offers some advantages for some applications, and there's a link here to that. Right, next, antibody isotypes, classes and subclasses. So these are the different immunoglobulin antibody isotypes or classes. IgG, IgA, which is a dimer or can exist as dimers. IgM, which can exist as a pentamer. IgD and IgE, which has this extra constant domain but lacks a hinge region. And the light chains are either kappa or lambda. And the heavy chains, well, the type of heavy chain dictates the isotype of the molecule. So if an antibody has a gamma chain or gamma chains, it's IgG. Alpha chain or alpha chains, IgA. So it's the correspondence of the Greek. So G, gamma, A, alpha, M, mu. So there are mu chains, heavy chains are mu chains. IgD, delta. So delta chain or chains. And E is epsilon. These are epsilon chains. And each B cell can only secrete one of these types. So all have the same Fab region, but differ in the Fc region of the heavy chain. And IgG, IgD, and IgE all have similar overall structures, although, as I said, IgE has an extra constant domain on the heavy chains and no hinge region. And IgM usually exists as a pentamer and IgA as a dimer. And these are the IgG subclasses. This is from Vidderson. And the Vidderson references, they really are excellent. So this is certainly worth looking at. So these are the four IgG subclasses: G1, G2, G3, G4. And you can see they differ principally in terms of the hinge regions, which connects the FC domain or region with the two Fab domains or Fab regions. So the hinge region will vary in terms of the length. So G3 has the longest length. On average, there'll be some variation with this through what we call different allotypes. Then IgG1, 15 amino acid residues, and then G2 and G4. And the number of disulfide bonds will also differ, with G3 again having the highest number of disulfide bonds, although IgG1, despite the fact it has a longer hinge region, only has 2 disulfide bonds. G2 actually has more disulfide bonds. Again, there's more detail in the Roy Jeffress lecture, which follows this one. And here are some properties of the different isotypes or classes. IgG, molecular weight, it's about 150 kilodaltons, although the G3 will be a bit larger than that because of its longer hinge region, maybe 165 to 170 kilodaltons. This is the major antibody in blood anyway, 90%. 4 subclasses, 1, 2, 3, and 4, with different numbers of disulfide bridges in the hinge region and different lengths. And generally at the long half-life of 20 days, although the G3, because of its longer hinge region, tends to be less stable than that. So GA, the molecular weight of the monomer, is slightly larger, about 160 kilodaltons, and is found in external secretions, such as the mucosal lumen in the alimentary and in the bronchial tracts. So it normally exists as a dimer, which takes its molecular weight, molecular size to be 320 kilodaltons on average. And this has 2 subclasses, IgA1, IgA2. And it contains extra peptides, the J chain, which links the monomers together in a covalent fashion, and something called the secretory component, and has a half-life on average of about 6 days. IgM, along with IgD, it forms receptors on the surface of immature B cells. It's found in serum as an approximate 900 kilodaltons pentamer. So it's the largest molecular weight of the antibodies, approaching nearly 1 million molecular weight. That's big. And the monomers are joined together to form the pentamer by the FC tails and J chains. Have a look at that diagram I just showed you. This is the first antibody to be produced in an immune response, and often has a low affinity but high avidity due to lots of low-strength binding sites. And IgM has an average half-life of about 10 days. And here's a reference explaining the difference between affinity and avidity of antibody molecules. IgD, very low levels in serum, as you just said, along with IgM, forms receptors on the surface of immature B cells. And binding of antigen to IgD elicits a signal transduction cascade that initiates B cell activation and proliferation involving T helper cells, as we discussed in the previous lecture. IgE, this is the main antibody of the allergic response. This has low levels in serum and interacts with the mast cells through its Fc receptor. The mast cells have a receptor specific for the Fc region of IgE on their surface. There's no hinge region, or not much of a hinge region, but an extra constant domain. So that means its molecular weight will be higher than that of IgG molecules. The binding of antigen initiates a signal cascade to trigger degranulation and release of histamine in the mast cells. Half-life, relatively short, about 2 days. And of course, IgE antibodies are responsible for allergic reactions. They have a cusp shape in solution, as we talked about before with the paper we did years ago with Dennis Burton's group. And a copy of that paper is on the Moodle. And also, you should look at the Sutton and Gould reference, which gives a very nice description of IgE and its binding to the Fc receptor, again with this cusp shape, which after we worked this out for using hydrodynamics, I think in 1990, was later shown to be correct from subsequent NMR and then crystallography structures on the IgE Fc fragment. Right, this table, taken from the immunobiology book, gives a nice comparison between the IgG classes and different isotypes. So there's a large variation in IgG isotype serum levels and half-life. And some functions are specific to particular immunoglobulins, and placental transfer is IgG specific. IgA, specific pathway of complement activation, again, which we talked about in the last lecture. And IgE has specific binding to mast cells and basophils, and is responsible for the inflammatory response. So I got the spelling mistake just there. And this compares the molecular weights. So IgE has a higher molecular weight than IgG. IG3, because of its longer hinge region, has a larger molecular weight, again between 900 and 1,000 and a million molecular weight for IgM. And because of its longer hinge region, IgG3 has a much shorter half-life compared to the other G subclasses. And this can be important, for example, when designing monoclonal based therapies using IgG in choosing the appropriate IgG subclass. Right, antibody diversity. Well, what gives rise to the diversity of antibodies? So we have a large range of antibodies for different purposes. And of course, we did a huge range to pick up the very large number, 10 to the power 8, I think, determinants or antigen determinants. Right, so first of all, each light and heavy chain has an N-terminal variable region and a C-terminal constant region. The light chain constant region dictates whether it is a kappa or lambda version, and the heavy chain constant region determines the antibody class. And the heavy chains can be gamma, it's IgG, alpha, IgA, mu, IgM, delta, IgD, or epsilon, IgE. And the gamma chains have 4 varieties. 1 gamma, IgG1, 2 gamma, IgG2, 3 gamma, IgG3, 4 gamma, IgG4, differentiating each of the IgG subclasses. The alpha chains have 2 varieties, 1 alpha and 2 alpha, differentiating each of the IgA subclasses. Again, this is this other Biddison reference, again, worth looking at, and this link is given here. First of all, these are the overall structures of the different subclasses. Again, you can see the variation in the hinge regions between the different subclasses, and this is shown in more detail here. And then we can get, this is the FC glycosylation here, linked to asparagine 297. And there's some variation in terms of the amount of fucosylation. For example, 94% in general is fucosylated, and then you have some differences along the chains here. The subclasses can also be glycosylated on the Fab regions, Fab domains, shown here. Not always. The glycosylation of Fab will vary between 5 to 30%. And at the end, you've got the sialic acid residues, which are hugely important. And you have these fucose residues. And this is described in much more detail in the Roy Jeffers lecture. It explains how differences in fucosylation and also other areas can have an important part in terms of the properties of these molecules. And maybe it's worth mentioning at this point, in the development of antibody-based therapies and monoclonal-based therapies against cancers, then one of the problems, and all the problems that the body's defense has, is that cancer cells are very good at screening themselves against antibody attack. So damaged cancer cells, which are normally removed, can be fooled by certain cancer cells because of the sugar coating on the surfaces. And most cells have sugar residues on their surfaces, but cancer cells generally have a higher degree of glycosylation, and largely due to sialic acid residues. So there's a lot of attention now is based in getting around this problem. I think I mentioned in the introductory lecture the great work of, for example, Caroline Bertozzi, and it's worth looking at her Nobel Prize lecture, which is on the YouTube from 2022. So glycosylation is very important, particularly the role of sialic acid and the fucose residues. Again, you can see more about that in Rod Jeffress' lecture. You can get also variations in amino acids, and also the hinge region, which for IgG, we call this allotype variation. Now, there are about 10 to the power 8 different variants of antibody, mostly due to differences in the variable region where the antigen binds. However, there are only about 30,000 genes in the entire human genome. So how does this variation occur? How can the armory of antibodies cope with the huge diversity of antigens that can be presented to the antibody defense system? Well, this comes from gene rearrangement and recombination in the variable region, and this was the video we showed in the introductory lecture, which showed the great work of Susumu Tanagawa in the immune receptors video, and it's worth looking at this again. And I think he got the Nobel Prize in 1987 for this work, and the video is quite old. It's 1986. But if you look at it again, it's only about 15, 20 minutes. It shows that he had this revolutionary idea, because it was previously thought that the genes were static structures and could not be rearranged. And his description in terms of gene rearrangement in recombination was against that dogma, but his experiments proved that's what happened in terms of the coding of the variable domain. And also this concept that one gene corresponds to one protein. And he showed that there were two genes, one coding for the constant domain and one coding for the variable domain. So these two dogmas had to be broken down before the biochemistry community accepted this, and now it's universally accepted. So look at that video again, but this is the essence of it. So the coding region for the variable domain on the light chain has these variable segments, a large number, not quite as many as was thought when Susumu was coming out with this theory. Gosh, it's only 40 years ago and now, but still a large number. So there are a large number of variable segments, and then there are these joining segments, and this is the segment coding for the constant domain. again, you can get some variations here. This is the lambda light chain. This is the kappa light chain. And the heavy chain has also, on the variable domain, has also these D segments, which corresponds to diversity or diversity segments. So there are 40 variable regions, variable segments on the kappa chain, light chain, 30 on the lambda light chain, and 40 on the heavy chain. There are no diversity segments on the light chains, and 25 on the heavy chain, and 5 or 4 on the light chains, and 6 on the heavy chain. And this is what happens. This corresponds to the light chain, but a similar sort of process happens with the heavy chain, but also including the diversity segments. So this shows the kappa light chain gene cluster on chromosome 2. And so there's 40 different B segments, 5 joining segments, and 1 constant segment. So as B cells differentiate, random recombination occurs on the DNA between 1B and 1J segment. And during mRNA synthesis, the remaining J segments are spliced out, rather like an intron. And similar recombination occurs for lambda light chain and the heavy chain, including the D diversity segments. As I said, the lambda light chain has 30 V segments, 4J and 4C. And the heavy chain has 51B, 6J, and 27D, and 9C genes. Gamma 1, 2, 3, 4, alpha 1, 2, mu, delta, and epsilon. We'll come on to this in a minute when we come to gene or class switching. So there are a very large number of possibilities in terms of combinations. Any light chain can combine with any heavy chain. So the final kappa light chain product has 40 times 5J equals 200 possible variants. The heavy chain has 51 V times 6J times 27D equals 8262 variants. So 8262 times 200 equals 1,652,400 variants of heavy and kappa light chain variants. And then we can get much further variation because the recombination stage can be imprecise, and this introduces further variation. So that's the basis of how we get such great diversity in the antibody variable region to recognize the huge number of possible antigen determinants. And we can get class switch via recombination. So on the constant domain, B cells can rearrange the constant region genes in the immunoglobulin heavy chain locus. So you can switch from expressing one class of IgG, such as IgM, to another during recombination, such as IgG, so a gamma mu switch, and this produces an antibody with different effector properties, because the different classes will have different Fc binding receptors without altering the antigen specificity. Okay, so you can find more information on these references here. So we've learned about the key features of antibody structure and how they bind to antigens. We've learned about the different immunoglobulin isotypes or classes and the different IgG subclasses. We've learned about the different mechanisms by which antibody diversity is established, including class switching. So next, we're going to consider in some detail IgG, its subclasses and glycoforms with the Roy Jeffress material. We're going to consider the aggregation problem in antibody biotechnology, how this is detected using what we call gold standard FDA approved technologies. That's focusing on the analytical ultracentrifuge. We're going to consider the pegylation of antibodies, which is one way in which the antibody problem can be addressed. That's with Mani, Dr. Nagamani Bora, and then with Dennis Burton from the Scripps Institute via live online broadcast from La Jolla in California. We're going to consider monoclonal antibodies. And you can see which is the best IgG subclass for particular applications and the connection with the development of vaccines. Okay, thank you.